Hello and welcome to our today's lecture of Introduction to Mechanobiology. So, over the last 3 classes, I had discussed 2 papers which link ECM stiffness with cancer. Okay. So, what these studies collectively demonstrated that when you have increase in ECM stiffness, so increase in ECM stiffness is driven by increased deposition of collagen 1, okay, and its cross linking by enzymes like LOX, okay, or lysyl oxidase. So, you remember earlier we had discussed how properties of collagen gel scale as concentration cubed. Okay. So, this tells you that increase in collagen density can trigger increase in bulk stiffness of the matrix, and when you have cross linking enzymes, then this stiffness can further increase. So, this explains the nearly 25-fold increase in stiffness of mammary gland from roughly 200 pascals to nearly 5000 pascals. Okay, you have a 25-fold increase in stiffness. Now, if you think of 3 dimensions, okay, on 2D, I can keep on making the matrix stiffer and stiffer. Okay. So, on 2D, the cells will sense the higher stiffness of the substrate, and this should drive, so increase in focal adhesion, okay, and increase in Rho-ROX signaling. Okay. So, these are 2 key regulators of cancer inertness, but the same cell, okay, if you think of a cell migrating through a 3-dimensional matrix, okay, when it is sparse, so in 3D, okay, what matrix provides is confinement and steric hindrance. Okay. So, in 2D steric hindrance is absent, but in 3D cells would have to contend with steric hindrance. So, in sparse matrices cells might actually pass through the pores. However, in very dense matrices, okay, when the matrix is very dense, migrating becomes a rate limiting factor, and in essence, as we will discuss later, nuclear deformation is the rate limiting factor in confined migration. Okay. So, how then with increase in stiffness, how do cancer cells become more invasive? Okay. So, that is the question, and it turns out that increase in stiffness leads to stiffening of the matrix, that leads to steric hindrance, but the cells have a way of dealing with that increased steric hindrance. So, what they do is they make use of degrading enzymes or proteinases which degrade the matrix, okay, thereby creating paths for migration. Okay. So, one of the most prominent family of endopeptidase is matrix metalloproteinase. Okay. In short, they are referred to as MMPs. Okay. And it has been observed that in cancer, multiple MMPs are overexpressed. Okay. So, in human there are 23 different MMPs. Okay. Some of these are matrix or membrane anchored. Okay. One of the major MMPs which are membrane anchored is MT1 MMP, is also referred to as MMP 14, okay. Or you have multiple soluble MMPs. Okay. Examples are MMP 2, 9, etc. Okay. So, one interesting bit of information is that there are many MMPs, okay, which co-localize with integrins, suggesting there might be some crosstalk between MMP activity with focal adhesion signaling. Okay. So, the finding that MMPs are overexpressed in the context of cancer would mean that if you inhibit MMPs, then it should lead to decrease or reduced invasion. Okay. And this idea led to multiple clinical trials. Okay. Multiple clinical trials which tried to target MMP activity. Okay. Unfortunately, all these clinical trials failed. Okay. And of course, there are multiple reasons for it. One of the most important reasons being MMPs are important not only for diseases, but they have multiple physiological roles also. Okay. They have important roles in development. Okay. And their activity is spatiotemporally regulated. Okay. So, they act in a very context sensitive manner. But there is one more reason which was demonstrated by Peter Friedel. Okay. So, the experiment that he did was he took cancer cells, okay, namely MDA MB231 and HT1080. Okay. So, these are breast cancer cells and these are fibrosarcoma cells. Okay. So, both these cells were highly invasive, and what Friedel demonstrated was when you inhibit proteases. Okay. So, generally you have cells which are migrating through these matrices, okay, by creating degraded zones. So, let us say this is a degraded zone. Okay. So, when you inhibit MMPs, okay, so when you inhibit MMPs, what he showed was these cells transition into more rounded cells, okay, where your matrix remains intact, okay, matrix remains intact, but they squeeze. Okay. So, this is mesenchymal migration, and they migrate in an amoeboidal manner. Okay. So, you remember from our previous lectures that we showed that, so this is protease dependent, mesenchymal is also called protease dependent migration. To protease independent type of migration. Okay. And this transition is called the mesenchymal to amoeboidal transition. Okay. So, here you have MAT. When you inhibit MMPs, the cells transition from adhesion dependent, protease dependent to adhesion independent, protease independent migration. Okay. So, this is so instead of path generating, okay, because proteases create path, you have transitioned from path generating kind of migration to path finding kind of migration. Okay. So, I had previously discussed that in amoeboidal migration, you can have multiple modes of migration in 3D, right, using more protrusion dependent or contractility dependent. Okay. So, the question which arose from all of these studies are all types of cells capable of exhibiting mat? Okay. And second is, what is the molecular mechanism which enables this switch? Okay. So, one way of seeing this crosstalk, okay, so you have crosstalk between MMPs and migration. So, what was also not clear is how is ECM stiffness regulating mesenchymal migration okay, and what are the phenotypic changes. So, what are the phenotypic changes associated with mat? Okay. So, to do this, one can do a simple experiment. You take two surfaces, so two polyacrylamide gels, one is soft and one is stiff. Okay. Let these gels be coated with collagen 1, okay, and imagine you plate 3 different cell lines, MDA MB231, HT1080. So, both these cells are highly metastatic. Okay. So, imagine you take another cell line MCF7. Okay. So, this is also a breast cancer cell line. MCF7 cells have been shown to be capable of forming a tumor, but they cannot metastasize or cannot spread. Okay. So, the experiment is you culture these 3 cells on soft and stiff PA gels which are coated with collagen. The stiff gel you can choose, let us say 5 kPa, which mimics okay, metastatic breast samples, breast tissue, okay, and this one is normal breast tissue, mammary gland. Okay. So, if you do this experiment, so what you can probe is, first of all, do cells exhibit a stiffness dependent spreading that we observe. So, what you would realize, so in terms of soft and stiff, if you take MDMB, okay, this is soft, this is stiff, again this is soft, this is stiff, okay, and this is your spreading axis. So, for MDMB and HT, what you observe is a dramatic increase in spreading between soft and stiff surfaces. Okay. So, there is an increase, but in MCF7 cells, these guys do not exhibit this stiffness dependence or much weak. Okay. What you can also do is you can assay the conditioned media. So, you can collect the conditioned media and then do zymography or gelatin zymography. So, gelatin zymography is a technique in which you have a regular page which has gelatin in it. So, you run the protein. So, you collect this conditioned media will have multiple proteins, including MMPs which are secreted by the cells. So, what this experiment revealed was, first of all, so from soft to stiff. So, when you run the zymography, what you see is, if this entire, so let us say you have two conditions, soft and stiff. You have these white patches corresponding to molecular weight of certain proteins, let us say MMP2 or MMP9. So, this would mean that in the gel, this is where this protein migrated, and when you activated the zymogram, these guys degraded the gelatin which was there inside these gels. So, causing a white zone. And this, by comparing the intensity of these white zones, you can compare the activity of this particular protein. So, what they observed was that in MDMB and HT cells, in MDMB and HT cells, so these cells ECM stiffness led to increased MMP 2 and 9 activity. So, in other words, these cells secrete more amount of proteases on a stiffer environment. So, this might explain why in a more dense network cancer cells are getting more invasive, because the increase in ECM density is triggering these cells to secrete more amount of proteases, which are then degrading the matrix, thereby creating paths for escape. So, this is the reason why on a stiff matrix or in a stiffer environment the cells are more invasive, because they secrete more proteases. Now, interestingly, this protease secretion is again myosin 2 dependent. So, if you inhibit myosin activity using plebistatin, activity of the protease activity is significantly reduced, suggesting that contractility. So, you have contractility modulating MMP activity. So, why must, what might be the possible reason in which this process is happening? Now, you know that on a stiffer surfaces, contractility leads to focal adhesion formation. Forces are required to stabilize focal complexes. So, given that MMPs have a crosstalk with focal adhesions, is it possible that there is some interaction between MMPs and integrins which is regulating this process? And so, for this, experiments can be done in which you treat cells, with protease inhibitors. So, GM 6001 is a broad spectrum MMP inhibitor. So, when you look at the spreading profile of MDMB on a stiff surface, so soft to stiff there is an increase, this is soft, this is stiff, but when you inhibit with GM, your spreading drops. So, this is plus GM condition. You have spreading increase, you add GM, it inhibits spreading, and what has been observed is when you treat cells with GM, this leads to perturbed integrin signaling. Okay. In other words, your focal adhesion phosphorylation levels of focal adhesion kinase 397 residue is dropping. Okay. So, when you had plus GM, this leads to inhibition of integrin signaling. So, how is this possible? So, to address it, you can actually look at integrin signaling experiments, integrin recycling experiments. So, imagine you have a cell, okay, and you look at, without permeabilizing the cells, you probe for integrins. Okay. So, no permeabilization, and imagine there are integrins which are sticking out like this. Okay. And of course, there are other of these inside the cell. So, if you treat cells with GM, what is observed is these integrins become recycled. Okay. In other words, all these integrin molecules, integrins exhibit increased cytoplasmic localization. When MMP activity is inhibited. Okay. So, if you take an antibody which binds to the extracellular domain, so what you would see, okay. So, if you take an antibody which binds to the extracellular domain, under control conditions, you would see a very clean red staining along the periphery of the cell, but when you treat cells with GM, you would see that in the periphery there is no clear localization, instead the entire protein gets localized inside the cell, suggesting, so this suggests that inhibition of MMP activity perturbs integrin stability, okay, at the membrane. Okay. So, what you have is a situation, okay, in which if you think of focal adhesions, okay, you know that contractility stabilizes focal adhesions, but also what you have is a situation, okay, when you inhibit with, okay, with GM, you add GM, your focal adhesions disassemble. Okay. Your focal adhesions, there is a drop in focal adhesions. This leads to loss of cell spreading. Okay. So, suggesting that you have a two-way crosstalk. On one hand, contractility forms stable focal adhesions, and this leads to robust MMP activity. But on the other hand, when you inhibit, so this is you are inhibiting MMP activity using GM, your focal adhesions fall apart, this is loss of cell spreading, and what this leads to is also loss of tractions. Okay. So, you have these results suggest that there is a robust crosstalk between contractility and MMP activity, where perturbation of contractility leads to loss of focal adhesions, thereby leading to loss of MMP activity. On the other hand, if you inhibit MMP activity, there is loss of focal adhesions that leads to loss of tractions and also leads to, okay, cell softening. Okay. So, could this be a mechanism whereby MAT is enabled? Okay. In one direction, Contractility is stimulating focal adhesions. Focal adhesions are leading to robust MMP activity. So, you have stable mesenchymal state. Okay. When you inhibit MMP activity using GM, focal adhesions are gone, integrin signaling is gone. So, integrin signaling is also gone, is perturbed, loss of cell spreading, also loss of cell motility. So, cells cannot move. Then leads to loss of tractions and loss of and cell softening. Now, these two things, if the cell becomes soft, can this be a phenotypic change, okay, which drives or which sustains the amoeboidal phenotype state? Because under amoeboidal migration case, your cell has to squeeze through pores. So, maybe this is the mechanism which enables cells to switch from a mesenchymal to an amoeboidal state. Okay. With that, I stop here. We stop our discussion about cancer. From in the next two lectures, I will discuss little bit about one example of atherosclerosis and one example from in muscular dystrophy, and we discuss how, again, what are the mechanobiological changes which have occurred as a consequence of the disease or are associated closely with the disease and sometimes drive the disease. Thank you for your attention.